past few decades, the research in molecular biology has achieved new heights with the advancement of science and technology. Venki Ramakrishnan, commonly known as Venki, is an esteemed Indian American and British structural biologist who has made significant contributions to this field. In 2009, he received Nobel Prize in Chemistry along with Thomas Stutz and Ide Yatnath for studying the structure and function of the ribosome, the molecular machine that synthesizes proteins in all living cells. He was born in 1952 in Chidambaram, an ancient temple town in Tamil Nadu. Both of his parents were scientists. After graduated from Maharaja Sayajirao University of Baroda, he moved to US to study physics, but later shifted his interest from physics to biology. Ramakrishnan was elected a member of the European Molecular Biology Organization in 2002, a Fellow of the Royal Society FRS in 2003, and a member of the US National Academy of Sciences in 2004. Ramakrishnan is also a Foreign Fellow of the National Science Academy of India. In 2010, he received India's second highest civilian honor, the Padma Vibhushan. In 2015, he was elected to a 5-year term as President of the Royal Society, an honor previously held by Sir Isaac Newton, among others. Throughout his career, Ramakrishnan has emphasized the importance of public engagement with science. He strongly believes about the importance of science education and the need to invest in scientific infrastructure. In addition to his illustrious career in science, Venki Ramakrishnan has also distinguished himself as an author. His 2024 release, Why We Die, The New Science of Aging and the Quest for Immortality, ventures into the evolving science of aging, questioning the inevitability of death and exploring the potential for extending human life. Recently, he has attended Kolkata Literary Meet, where he discussed the science and philosophy of immortality, how science shapes literature and how storytelling can enhance scientific writing.
Welcome. Today we have with us Dr. Venki Ramakrishnan, who is a very eminent scientist who works at the MRC Laboratory of Molecular Biology in Cambridge. Dr. Ramakrishnan got the Nobel Prize in Chemistry for his work on ribosomes and unlocking a very basic mystery in the way our body works. And we're going to talk about his trajectory in scientific education and research, and also talk about his current work on research in the science of aging, for which he's written a wonderful book which came out in the last year called Why We Die, and he is here in Kolkata today to talk about both science in general and also about what he's written in this amazing book. Welcome, Dr. Ramakrishnan. I would start by asking you about your career. Starting off, it's very, very interesting way you moved from growing up in India, in Baroda, studying physics. Of course, at that time, at the University of Baroda, physics was taught in a very, very interesting way. I wish you could talk about how you got into sciences in physics and then moved to do a PhD in physics in the US, and then moved on to take up biology as your subject of your life. And it's very important for people who are now listening to you to understand how one can move through a very unconventional PhD and research career, whereas most people in India, go into engineering education nowadays, away from the pure sciences. and to then chart how you moved into this very, very interesting career that led you to the Nobel Prize.
Yeah, so I'm afraid it wasn't a lot of intelligent planning on my part. I think I'm not sure I would recommend my career path to anyone, because I could have fallen off the map at one of many different stages. It happened because I graduated from high school, and in those days you had to do a pre-science before you then went on to your career, which could be engineering or medicine and so on. And my parents were academics, and they were not big believers in all these coaching classes for national exams. And I just took the IIT exams on a whim without really preparing for it. In fact, I couldn't have prepared for it because our university was very progressive. It had a completely different curriculum. They taught something called PSSC physics, which was a very modern way of learning physics, but it didn't really prepare you for these IIT exams. And needless to say, I didn't do very well. I didn't get into any of the IITs. I also tried to get into Vellore Medical College and didn't get into that either. And so
PSSC physics, was it based on some unconventional textbooks? Or
it was an unconventional textbook. It was written in the US but republished in India and adopted by some progressive universities, including mine. And it was a very empirical way of learning physics by doing and observation and some very interesting concepts. So anyway, I took all this, and I didn't get into the IITs. I didn't get into Vellore Medical College. I got into the Baroda Medical College and the Baroda Engineering School simply because I did very well on my pre-science. So if you were in the top 10 or 20, you automatically got admitted. But then my mother made me not made me, but I suppose she encouraged me to take the National Science Talent Scholarship exam. And this was done as a government program to encourage students to go into basic sciences. And it involved doing a science project and then also taking a written exam and then an interview. And oddly enough, I was one of 4 in my state, which was Gujarat, who were selected for this scholarship. And so I thought about it, and in the meantime, what had happened is a number of faculty members in Baroda had come back from the US, and they'd completely modernized the physics curriculum, and the mathematics curriculum had been similarly modernized. And so I had the chance to study things like the Feynman lectures on physics and the Berkeley physics course as part of my undergraduate training. So I thought about it and thought, well, maybe this is a sign. You know, I didn't get into these top engineering and medical programs, but I got this top scholarship, and maybe I should do basic sciences.
Because science benefited from that.
I guess so, eventually, maybe. But anyway, so I took that, and I studied physics, and then normally what would happen is people who wanted to go abroad would do a master's in India before applying to the US or to Europe. But my father was doing a short sabbatical in the US, and I was somehow eager to get out and move, explore the US. I was 19 years old when I graduated, which is somewhat younger than usual. And so I was eager to go to the US. So I applied to a bunch of schools at the last minute, but I hadn't taken the GRE exam. I didn't have a master's. And so I went to the only university that would give me a fellowship, which was Ohio University in southeastern Ohio. And so there I was, off to the US at the age of 19 to do a physics PhD. And
did you get to choose your subject within physics, or?
Yes, you could. You spent the first 2 years taking, so it's not like the UK where you have to choose an advisor right off the bat. In the US, in physics departments, typically you take a year or two of courses, then you take a comprehensive exam, and then you settle on an advisor. And I had no trouble taking the exams, and physics departments are ruthless about weeding out people in these comprehensive exams. So for example, in my batch, only about 7 out of 19 made it. Okay, and the others had to either repeat it or drop out with a master's degree. Anyway, so I passed the exam, and then I realized I didn't have any sense of what research in physics actually meant.
And this was in theoretical condensed matter physics.
That's what I ended up choosing, but you could have asked me about any area of physics, and I was completely ignorant of what was going on in contemporary physics. I was very knowledgeable about what I was taught, quantum mechanics and classical mechanics and electrodynamics, things like that. But when it came to research, I was completely lost, and I settled on a Japanese professor named Tomoyasu Tanaka, who was a condensed matter theorist. And in some ways it was a misfit. I mean, he was good at his field, but I'm not sure he was able to convey to me a clear idea of what the real questions were and what to do. And I, on the other hand, wasn't thrilled by the problem, and my interest was drifting, and I was getting into all sorts of other things, like hiking and playing chess and so on. I was goofing around, and he was too polite to tell me to shape up. And so this continued for a while, and in the meantime, I started looking at, I used to read Scientific American, and what I found was that physics is a mature field, and the really big questions are very, very hard. Whereas in biology, it seemed like it was exploding, a bit like physics in the early 20th century.
How did you come across biology in the first place? I mean,
in Scientific American.
Aha. Yes.
So I would read all these articles, and I would say, this seems to be where a lot of the action is.
In those days, Scientific American had actually the scientists writing articles
Yeah,
themselves.
it's a very different magazine
very different
now.
from now.
Scientific American in those days was superb because all the articles were written by scientists. They had great help from the editors to make it readable, but it was primarily written by the scientists talking firsthand about their work. And if you look
I remember
back,
Stephen Hawking wrote his Hawking radiation paper first in Scientific American before publishing it.
It was just a spectacular magazine, and I'm really sorry at its evolution. It has become a kind of, almost an ideological rag, I have to say. And I hope, now the editor's resigned and they have a new editor, and I hope it improves as a result. I was exposed to all this biology, and I thought, this is where I ought to be. And I knew that many people who had started off in physics had made it big in biology. For example, the most famous one is Francis Crick. But there's also Max Delbruck, who was actually a well-known physicist before he became a phage geneticist. And so I thought, well, okay, this is something I should do. And I applied to several graduate programs. And many of them wouldn't admit me because they said, you already have a PhD. But a few did. And so I chose to go to the University of California in San Diego, which had a very broad and large biology program. And arriving there was also interesting because when I went there, I couldn't understand any of the introductory lectures, research talks that these faculty members gave.
Undergraduate level, right? So, or even.
So I was admitted as a graduate student. So these professors would give little research seminar talks about their work to introduce graduate students to what was going on in the department. And I couldn't understand a thing they said. So I went to my advisor and said, look, I'm afraid I need to go even further back. And so the first year I took all the undergraduate courses I could to come up to speed. And then the second year I started doing some research in biology.
You kind of started all over again.
You have to. I think when you go to a new field, you have a choice. you can either continue to do what you did but simply try to apply it to the new field, then you'll be sort of a niche player. But I thought it would be better to get a broad understanding of biology, to ask what were the questions. In other words, I wanted to become a biologist. I didn't want to be a physicist doing a little bit of biology.
And then, so that worked out. And in the end,
It worked out in the end, but not for a long time. For example, after 2 years of graduate school, I went and did a postdoc at Yale to work on some aspect of the ribosome. And one reason I think I was given that postdoc is they were using a physical technique called neutron scattering.
Which you knew as a physicist.
Well, which I didn't know, but I could easily pick up with my physics background. And but it turned out that neutron scattering was a dead end in terms of its importance in biology. It's always remained a niche technique.
But it's interesting how these ingredients come together to carve your path. You couldn't really predict.
Sure, but for example, after my postdoc, I applied for 50 or so faculty jobs, and I didn't get a single interview, right? So that's another point when I could have just simply fallen off the map and done.
And that's because of your unconventional background. People were looking for people who've done biology for a long time.
That's right. Here I was, with a physics degree from a not a major, top-ranked university, and then I had done 2 years of biology but no degree in biology, and then I'd done work on the ribosome using this weird technique that they didn't know about, and so of course it didn't make any sense in terms of fitting into their department. And I also applied to 4-year colleges, but they probably saw my long Indian name and they thought, well, we don't even know if this guy can teach, does he speak English well? So I think for all sorts of reasons, I could have followed.
What prompted then your move to the UK?
I was finally managed to get a job at Brookhaven National Lab after an aborted year in Oak Ridge, which was another national lab. But at Brookhaven National Lab, I published enough using neutron scattering and some other techniques to be given tenure. But then I realized that was sort of the end of the line for what I could think of with neutron scattering. And it really wasn't the technique to answer big questions. So I told my tenure committee, they asked me, what will you do if we give you tenure? I said, well, the first thing I'll do is I'll go on sabbatical and learn crystallography. And because that's the way to get atomic resolution structures of large molecules, and that really connects genetics with chemistry and function. And that's where the action is.
By the way, that's where Crick came to biology as well, through crystallography.
Exactly, and in fact I went to the same institute.
Yes.
So the institute, which you mentioned, the MRC Lab of Molecular Biology, was started by Max Perutz, who also had a physics background from the Cavendish, and then Crick was his student. And so there were all these connections. And so I ended up writing to Aaron Klug, who was also a physicist turned biologist, who was the director of the lab, and also went on to be the president of the Royal Society. And he could have easily thrown my application into the waste paper basket because I was a complete nobody at the time. But I had crystallized a protein that he was interested in. And I told him, look, I want to come there and learn how to solve it. And so I want to spend a year there learning crystallography. And he sponsored me for a Guggenheim Fellowship. And that's how I got to Cambridge on sabbatical. So I learned crystallography that year. It was very successful. Came back, and I actually had 2 Nature papers from just that 1 year sabbatical, using data we had collected at Brookhaven. So I came back, and then something about the MRC struck me, which is it was a place where they never bothered with trivial incremental stuff, but it was always directed towards some long-term goal, which was an important problem in their area. They could do that because the place was funded by the MRC and had relatively stable funding. So what I did is when I came back, I was a little unhappy, and DOE, which ran the national labs, also started being more interested in highly applied, large programmatic work rather than this bottom-up basic science that is more typical of university labs and is typical of the MRC. So I was looking around. There were no jobs at the MRC lab at the time. So I went to the University of Utah, which had a superb group of academics, including somebody who went on to win the Nobel Prize a few years later, many National Academy members and so on, and particularly strong in my field of RNA and structural biology. And I also liked the environment. I like hiking and the outdoors, and so it was a beautiful place. So I went there, and but then my sabbatical here at the MRC had made me ask, what is a big question in my field? And the big question in my field was, if you want to understand how genetic information is translated to make proteins, it's done via this enormous complex molecular complex called the ribosome. And the ribosome had been discovered in the late 50s, but nobody knew what it looked like because it was too large, and so people had not solved its structure. And so my experience in crystallography and my year at the MRC made me think, actually, with the current methods that are available with these powerful sources of X-rays called synchrotrons and modern computing and so on, it ought to be possible to solve the ribosome.
Well, that's fantastic. I mean, just for the sake of the audience, I mean, of course, a lot of people know that the DNA actually encodes a lot of the information about the body, about our biological system, but then it is transferred to what is known as the RNA, which is responsible for protein synthesis in our bodies. But now the missing link between the RNA and the synthesis process is this very, very enigmatic system called the ribosome, and so you cracked it.
Well, a number of us did, but anyway, so I could have done it in Utah, and in hindsight, it would have, at least the first stage of it would have worked out in Utah. But then I thought, ribosomes had first been crystallized by a German group, group in Germany, by an Israeli woman, Ada Yonat, and the director of the Max Planck at Berlin named Heinz Günther Wittmann. And Ada Yonat had been working on the problem for a long time, and it seemed to us that she was stalled, and there'd been no progress for quite a while. And I've written about this in a previous book called Gene Machine.
The first book was about your work on the ribosome.
It was sort of a very frank, gossipy memoir of this race for the
great anecdotes
for the structure of the ribosome. So anyway, a number of us, after listening to Yonat speak at a couple of meetings, we thought, well, maybe this field could use other participants and some fresh ideas and so on. And I had my own ideas based on the fact that you can tune synchrotrons to any wavelength of X-rays, and that allows you to use a particular technique in crystallography. And so based on that, I thought maybe it's time to crack the ribosome, but I didn't know for sure whether it would work and how long it would take to work. And one problem with universities is they're dependent on grant funding. And grants are typically 3 to 5 years. And Yonat had been working for 15 years by this time. And so I thought, well, maybe I should write to the MRC lab where I'd done a sabbatical and find out if they would be interested in supporting this project, because it's the sort of thing they would like, a big fundamental question in biology. And I wrote to Richard Henderson, who was the director of the lab, and suggested this idea, and he asked me to stop by. I was going to a meeting, oddly enough, in Sweden, and I stopped by in Cambridge on my way there, and we discussed a lot of the ideas, how I would go about it, what the competition was like. and so on. It was a very intellectual discussion. He never discussed how much space he would give me, equipment, salary. None of that was discussed. It was a very odd sort of interview, if you like. First of all, there was no job. So it was a job interview without a job.
What do you like? And what do you do?
It was a job interview without a job. But anyway, after that, he wrote back and said they thought it was a good idea. They didn't have any space, but they would get in touch as soon as there was some. So I having lived in America for most of my adult life, I just thought this was a polite rejection. But actually, a couple of months later, he said, well, the space actually has materialized now. So yes, we're ready to hire you. So what do you think? And this suddenly made me go into panic, because it's one thing to sort of explore it. Now I had to commit to it. And I had to leave the US, where we had 2 grown children, and I had to take about a 50% salary cut and leave a place where I had very good colleagues and where I liked the lifestyle, and go off to Cambridge on this whim.
And from the mountains to the flatland.
I thought a lot about what we should do, and then I realized this was a once-in-a-lifetime opportunity. And if I didn't sort of stop everything else and go for it, I might watch it happen, done by others, and then I would regret it. But if I gave it a shot and it didn't work, I would feel, okay, I gave it a shot. And so I asked my wife if she'd be willing to move, and she had liked her sabbatical year, but she said, look, I'll move this time, but it's the last time I'm going to move. And in fact, she's been true to her word. We can't move from Cambridge now. So anyway, we packed up all our stuff and moved. And then after a year of really very stressful problems, lots of technical problems which prevented us from getting to high resolution, we eventually were able to manage to overcome that and get a high resolution structure of this small subunit. And the large subunit was done by 2 people at Yale, Tom Stites and Peter Moore. And Peter Moore was actually my postdoc mentor. So it was very interesting competing with a group who were your mentors.
They're working on 2 different scales.
We're two different things, but it still slightly mattered who came first. And in the end, it was sort of a tie. And the three of us, including Ada Yonat, who had, of course, started the crystallography on it.
But can I ask you there? I mean, we were talking about Crick and Watson, and the most famous story of double helix and the DNA, and the fact that the equipment and the method was already there because of Bragg's work, and so in science in general, you find the discoveries are made when the time is right, the technology is right.
Definitely.
So was that the case here as well?
Absolutely.
Something had come together which was new? Yeah.
Because in the end, about 3 or 4 groups published X-ray structures of the ribosome after a 15-year hiatus. So why is that? Well, one is that competition is good for science. It's not good for scientists because it's very stressful. But if you don't have competition, you tend to be complacent. You don't really work quite as efficiently and as fast and think as hard. So competition actually is not detrimental to science. People, there's an ideological thing that, oh, you should all collaborate. It doesn't always work that way. Sometimes collaborations are essential, like the LIGO experiment we were talking about, but sometimes competition is also good. So it's like anything else in life. But the other thing that did make a difference was that synchrotrons came online, very good detectors like CCD and then pixel array detectors.
It was just the time when the digital detectors came.
And so you could measure data very accurately. And then computing, also including computer graphics to look at these enormous molecules, also became more powerful. If it had been 10 years earlier, the computer graphics wouldn't have been able to even display a lot of the ribosome.
It had the spectral information, it also had high resolution information, images.
Yeah.
And of course now, I mean, so for example, this was, what, 30 years ago?
Yes, 25
years ago. And now, of course, there's more technology that's come on. I mean,
Yeah,
if you look
so
at
I should-
microscopes and the image technology and with artificial intelligence or whatever,
Yeah.
would you do it differently now?
Nobody would solve the ribosome the way we did.
How would you do it now?
So now the
If you didn't know
method of choice was developed, and one of the developers was the person who gave me my job in Cambridge, Richard Henderson, who won the Nobel Prize in 2017 for cryo-EM, along with two other people. So the method of choice today is a technique called cryo-electron microscopy. And here what you do is you take ribosomes in solution and you rapidly cool them down to liquid nitrogen temperature. So they're not frozen like ice, but they're in a glass-like state. And then you take microscopy images of them, and there are very clever algorithms developed by a number of people to reconstruct the three-dimensional structure from these particles. Now, this method had been around for quite a while. In fact, even when I started doing the crystallography of the ribosome, people like Joachim Frank had actually produced some really decent-looking images of the ribosome.
It would have been very model dependent though at that
No,
time.
it wasn't model dependent, but it was very low resolution. So it was sort of 25 angstroms. And so we crystallographers used to sneer at it as blobology. But actually, Richard Henderson had the foresight to realize, and in a seminal paper, he showed that actually electron microscopy had the potential to produce atomic resolution images if you could improve the microscope and the detector. And that's exactly what happened over 15 or so years. So by 2012 or 2013, very fast electron detectors came online, and the microscopes had improved, and new software that took advantage of these fast detectors to do motion correction came online. And as a result, these blurry images suddenly became much better resolved images. So today, probably an undergraduate project student could solve a ribosome structure in a summer using electron microscopy. And it probably could be done in 2 weeks by an expert.
But with this state of hardware, and hardware by I mean not just the microscopes, but also the back-end computer hardware.
Yeah, and the
with
software.
And then the software, which relies on this hardware. What's the next step?
Well, now the thing you can do, there are two advances. One is that with electron microscopy, you can not only get one state, but you can get a series of states, which you would only be able to do occasionally in crystallography and only with great difficulty, because you'd have to purify each state and be able to crystallize that. And these states are not always stable. You know, they can be transient and so on. So X-ray EM, cryo-EM, is giving us a much more dynamic snapshot of what molecules are like, how they're moving, and so on. And that's changing things. The other is that people can look at not very high resolution, but reasonable resolution images of molecules inside cells. So you can not only get high resolution structures by cryo-EM, but now you can also see where these molecules, how they're distributed inside cells, what is their organization, and so on. So what's happening is we're going all the way from chemistry to molecules to cell biology and their actual function in the crowd of the cell. And so that's a big excitement. The other is there are computational methods now to calculate structures of molecules. And this is because it's been known for a long time that the information on how a protein folds up into a three-dimensional structure is actually present in the sequence. So a protein is made, it's a particular sequence of amino acids. That sequence is dictated by the information in our genes. So the order of the DNA letters is what dictates the order of amino acids. And that in turn, the chemistry of it makes it fold up into a unique structure. what people had trouble doing is predicting that structure from the sequence. And there have been lots of computational methods, and they were worked okay for small proteins. But then people like Demis Hassabis and later his colleague John Jumper at DeepMind, which is a company that Demis Hassabis founded. And they decided to tackle this protein structure problem. And I wouldn't call it the protein folding problem, because they don't tell you how the protein folds. It tells you what the final structure should look like. And to my astonishment, a few years ago, they showed me the results of their latest version. And it was amazing how accurate it was in so much of the time. There were still some outliers where it didn't quite work, but I could see right away it was going to be a game changer. So what is happening now is that people are doing electron microscopy. Not all of the parts are highly resolved so you can interpret it in atomic detail, but even if it's low resolution, you can take an AlphaFold model. AlphaFold is the name of this structure prediction program, and fit it into the density and see that it actually makes sense. And so it's revolutionized structural biology. The other thing is that cell biologists and biochemists now who are looking at new proteins can make an AlphaFold prediction, see if it makes sense, and then test it. They can test the structure, they can do mutations, they can see how it might work, and then test it experimentally. So again, it's been a big game changer, and it's why, of course, that Hassabis and John Jumper shared the Nobel Prize, along with David Baker, who did the flip side of it, which is he said, if I want a certain structure, what is the sequence I need to make that structure? That's called protein design. And that also has huge applications.
So this year, of course, as our audience members know, that there were elements of AI and machine learning that dominated the Nobel Prizes, both in chemistry, as Dr. Ramakrishnan just said, but also in physics, where elements of AI came in. There are quite a few Indian origin Nobel winners now who have got their Nobel Prizes working outside India. You have economists, you have scientists.
Yeah, not so many in what I would call the natural sciences.
Not so many in the natural sciences. Yeah. However, the last natural science Nobel Prize that was given for work in India was the 1930s, right? I mean, C.V. Raman. But then Indian science has produced amazing results, mostly in the theoretical sciences, and many in experimental sciences, and we see very visibly a very large number of Indian origin scientists all over the world doing very well. What do you think would it take for Indian science to produce Nobel winners working within India? And why is it not happening?
I think, even in the West, there are lots of Indians who are doing extremely well, but there are, as you say, very, very few who've, forget about the Nobel, but let's say received major awards, who are considered clear leaders in their field, because the prize is somewhat arbitrary. But I think, well, one person in India who's received a major award is Ashok Sen, who won the Breakthrough Prize for work done in India. I think part of the reason people like him don't get considered for the Nobel Prize is because string theory is still a theory. It's very hard to, there's no good empirical way to verify it. Pre-independence, you mentioned one Nobel Prize, which is C.V. Raman, but there were other physicists of similar stature. For example, Meghnad Saha or S.N. Bose. Or you could say S.N. Bose was maybe a one-trick pony, but Saha did a number of things, and also J.C. Bose was really a polymath who did a number of different things. So there were lots of people in that caliber. The question is, are there Indian scientists who are world leaders in their field? Forget the Nobel Prize. Are there people who are world leaders in their field? And it's not clear that there are many. There might be a few, but by and large, many of them do what I would call really good work, but not world-leading work. And I don't know what the problem is. I think it could be also a problem of mentorship and institutional tradition. For example, why does the MRC Lab of Molecular Biology produce so many Nobel Prizes? And I think part of it, there are two reasons. One is when you go there, you're expected to not work on a pedestrian problem. You're expected to try and tackle something important.
And you're encouraged.
And you're encouraged to do that. The other is that when you do that sort of problem, you have a number of difficulties. One is, are you picking a problem that's premature and it's not actually solvable at the current time and you're going to waste your time? So that's one problem. The other is, let's say you choose a problem and obviously things are not going to work right away. The question is, should you keep going or should you give up? Or should you keep going but change your approach to a completely different method or a different attack? These are not things that you can always figure out on your own, but if you're surrounded by people who've done very difficult things themselves, they can give you feedback. They can say, look, this problem is not doable right now, you should do this other thing first. Or they could say, actually, I think you're wasting your time with this approach, why don't you try this other approach?
You know,
so that sort of mentorship is why often very good science is concentrated in a few places. If you go to Britain or the US, it's not everywhere that you have really top-level science. A lot of first-rate Indian institutions would be fine. They'd be fine in many parts of the US. It's just there are often institutions which have a tradition of excellence.
Do you see the same phenomenon playing out in say sports? I mean, Indians don't win Olympic medals and the level of mentorship is not there. And
exactly, and I'll give you a counterexample to that.
Yeah.
For example, Vishwanathan Anand grew up in a sort of vacuum. There were no top-level chess players in India, but nevertheless he made it to the very top of the chess world. But then what he did was he set up a chess academy in Chennai. And the result of that is today there are a large number of grandmasters who are Indian, including the current world champion, right? So that's a clear example of where top-level mentorship and training can be helpful.
So are you aware of the new education policy that now the Indian government is adopting, which opens up education to interdisciplinarity, makes things a lot flexible, and to bring in research into universities, which was one of the major problems of Indian research.
Yeah.
Do you see that helping?
Yeah, I think interdisciplinarity is a big buzzword, and people think of me as an interdisciplinary scientist, but actually I don't think of myself that way. I think of myself as somebody who wandered from one field to another and then simply used the tools that were best for the process. For example, Tom Stites started life as a chemist and ended up as a chemist, and he did exactly the same sort of work for which we shared the prize. So I think interdisciplinarity should be encouraged, but can never be forced or artificially made compulsory or something like that. But on the other hand, the other thing you mentioned, which is research and education, the formation of these ICERs, which combine undergraduate education as well as graduate education with research. I think that's a very good step.
I always thought that one of the major problems of Indian research was that they were confined in research institutes, so the younger people didn't see the benefits of it.
Sure.
And once the young people start seeing research happening around them and are taught by research active professors, it's going to go very different. I think we might be running out of time, so I wanted to come back to the book that you've recently written and have a very short discussion on the book, which is called Why We Die, the new science of aging in the quest for immortality. And this came out within the last year and has caused quite a stir. This book is absolutely fascinating because it looks at various aspects of aging and death. One of the most fascinating parts of it, of course, is that you are not confined to the biology of aging and death, but also you looked at the sociological and philosophical aspects of death, and maybe
only in the last couple of chapters.
And the last, so let's talk about the last couple of chapters, where you have gone into various ways of looking at how different cultures react to aging and death, how they react to the aging population, and how one confronts death. I mean, the expectation from death in certain cultures is that there's a life after death. In many other cultures, death is the end, and in many other cultures, coming back as something else is thought to be a good thing. So was that something that drew you into thinking about this, or you learned about this as you wrote this book?
No, I thought about it in the course of writing the book, but that wasn't really the purpose. The purpose of the book was really to discuss all of the big advances in our understanding of the biology of aging that happened over the last 50 years or so. So that's really the biggest part of the book. But then I wanted to ask, well, supposing all these methods actually work out and we all end up living much longer. For example, if all of us started living to be over 100, for example, or in extreme cases, if you can even break this natural barrier of 100 or 110 or so that humans have, what if we all start living even longer than that? What would it do to society? And I think that was the question I wanted to address. People have accused me of stepping outside my area of expertise and practicing amateur sociology or prediction. But
that is the point of writing a book.
Yeah.
And I think.
But I felt I was entitled to at least a discussion.
But of course, you've connected very nicely with the whole point of the biology that leads to aging. Because inherent in this problem of living long and what it does to society is also how feasible it is. There's no point living for 200 years if my organs don't work or my cognition doesn't work.
That's true, but the goal of the research community is not simply to extend lifespan, but to extend healthy lifespan. I mean, that is a primary goal. And the feeling also is that if you tackle the problems of aging itself, then you will also delay the onset of many of these diseases, because many of these diseases are a result of aging. For example, if you take the 4 main causes of death in old age, which are heart disease, diabetes, dementia, and cancer, all of them are correlated with age. So the highest risk factor, you could argue, is age, because whatever regulatory system that keeps these things normal is breaking down with age. And so if you were to slow down or attack many of the causes of aging, you might also postpone the onset of these diseases, so you would probably stay healthier longer. The question that is still unanswered is, what happens at the end? Will you suddenly die, or will you still have this long, slow period of decline where things are gradually breaking down? That's not a solved problem. And the current data suggests, unfortunately, that while our lifespan has increased, so has the fraction of our lives that we're unhealthy, the number of years that we spend unhealthily. So it remains to be seen.
Well, thanks very much, Dr. Ramakrishnan. You gave us a lot of things to think about. We learned of a very, very unusual career in science that went from India to the US to the UK across different subjects of physics, chemistry, biology, and led to a very, very successful career. Thank you very much for being with us tonight. Thank you.